Yesterday we have seen two high yield reactions in glycolysis. One is first reaction, conversion of glucose to glucose 6-phosphate done by glucokinase and hexokinase. And the second high yield reaction is PFK1, which does conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. Now, glucose is a 6-carbon molecule. Hexose means 6 carbon, right? Fructose is 6 carbon. And then fructose 6-phosphate, fructose 1,6-bisphosphate, all those are 6 carbon. And then if you carefully look at this part of the figure here, which I want you to know for further understanding, is fructose 1,6-bisphosphate, which is a 6 carbon molecule, breaks into 2 molecules now. It just breaks into in the middle, you break and divide into 2 molecules. And that is dihydroxyacetone phosphate. And the other is glyceraldehyde 3-phosphate. And now the way this figure it is written here, although they have shown dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, in that glyceraldehyde 3-phosphate, they have written number 2 here. What that means? You have 2 molecules of glyceraldehyde 3-phosphate. The meaning of this is the dihydroxyacetone phosphate, which is made after breaking fructose 1,6-bisphosphate, is in fact converted into glyceraldehyde 3-phosphate, making 2 such molecules. One, originally you get glyceraldehyde 3-phosphate. Another molecule of glyceraldehyde 3-phosphate comes from conversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate. So that means technically from one molecule of fructose 1,6-bisphosphate, you got 2 molecule of glyceraldehyde 3-phosphate. Remember this. Okay. And from now on, all these rest of the molecules, they have written it as 2, 2, 2, 2. That's it. Whatever goes on from that point onwards, from that glyceraldehyde 3-phosphate onwards, everything that comes in goes out. It is all 2. That's how your calculation is. And that is what is written in the figure. Yes. Correct. Yes. You break it into 2. You know, your fructose 1,6-bisphosphate, it breaks into 2 molecules, 3 plus 3. You know, each of these 2 molecules, 3 carbon. 3 carbon dihydroxyacetone phosphate, 3 carbon glyceraldehyde 3-phosphate. Later, dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate, making 2 such molecule. At that time, you don't have dihydroxyacetone phosphate because it is converted to glyceraldehyde 3-phosphate. That's our understanding. That's how you need to read this figure. Okay. So, with that understanding, glyceraldehyde 3-phosphate, the one that we got from breaking down of fructose 1,6-bisphosphate. This is done by enzyme aldolase. We will use that enzyme name later. There is an importance of that enzyme in the next PowerPoint. There is a disease related to that. I will come to that point later. Right now in glycolysis, that's not important, right? So now glyceraldehyde 3-phosphate. So 2 molecules. Remember, we are looking at this from this point onwards. It is all 2 times it will go on. So glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. That is done by enzyme. It is there in the name. Glyceraldehyde 3-phosphate. That's a substrate. Dehydrogenase. And dehydrogenases, they catalyze oxidation reduction reactions. That means your glyceraldehyde 3-phosphate is oxidized and NAD, which is entering into the reaction, is reduced into NADHH+. It's an oxidation reduction reaction. So NAD is getting in and NADHH+ comes out of this reaction, while your glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglycerate. Enzyme name is glyceraldehyde 3-phosphate dehydrogenase. Okay. And also you note here that inorganic phosphate is getting into the reaction and it will be attached to the first carbon of glyceraldehyde 3-phosphate. That's why the name of the product 1,3,1,3 biphosphoglycerate. There are 2 phosphates. One is at the first carbon, other is at the third carbon. Okay, that's your product. This is the reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase. Okay, and if you carefully look at the product, especially the first carbon, the way the phosphate is attached, look at the symbol here. The symbol, that's a special symbol that is shown there. And that symbol always indicates high energy phosphate bond. Whenever you see that symbol in a molecule, that is where is high energy stored. And if you break that phosphate, it's an exergonic reaction. There is a potential to generate ATPs if you do that. And that's what happens in the next reaction. When you break open that bond, remove that phosphate, so your cell will be able to make ATP using that. Okay, that is what is written, high energy acyl phosphate bond. Okay, so that means in this reaction, there is a high energy molecule that is generated, and that high energy molecule is 1,3-bisphosphoglycerate. So glyceraldehyde 3-phosphate dehydrogenase, it's going to oxidize glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, which is a high energy molecule, where NAD is needed in the reaction, NADH comes out. Okay, now this enzyme has got thiol groups. Thiol group come from which amino acid? SH group. Which amino acid has got SH group? Cysteine, amino acid cysteine got SH group, thiol group. So this glyceraldehyde 3-phosphate dehydrogenase enzyme has got thiol group, and arsenate is going to compete with the inorganic phosphate, that inorganic phosphate which is trying to bind to the thiol group to conduct the reaction. So whenever there is arsenate in place of in inorganic phosphate, arsenate will go and bind, and thereby do not allow this enzyme to function properly. Although it is written like competes, arsenate competes with inorganic phosphate, don't get confused with the competitive mechanism, because the competition is not with the substrate, that is glyceraldehyde 3-phosphate is a substrate for the active site, right? In a competitive inhibition, the inhibitor competes with the substrate for the active site, whereas here it is competing with the other molecule, that is phosphate, not for the active site, for the other binding site. And thereby it causes irreversible inhibition of the enzyme. And that's a non-competitive mechanism. So arsenate, it is, although it is written competes with inorganic phosphate, but don't get confused for competitive mechanism just because of that word. Okay, you got to be careful. Always remember, arsenate or any heavy metal ion, what kind of inhibition they will have on the enzyme? Non-competitive. Just remember that way. Arsenate, lead, mercury, whatever heavy metal, if they have any inhibition on the enzyme, you just go for non-competitive. Okay, so with that, let me, I have written already non-competitively just to make sure you know it. So before I move on, which vitamin is necessary to conduct this reaction so that this reaction goes on optimally? It's a niacin, NAD, look at NAD. So that's niacin. So in glycolysis, this is the reaction where you need niacin. Okay. So niacin, what can be the side effect of niacin when you take it at higher doses? Anyone take a niacin? Niacin supplements. Vitamin B3. The side effect of niacin, side effect in the sense like basically its effect. What it does is it causes vasodilatation. Niacin causes vasodilatation, allow blood to gush into peripheries. And that's why you feel bit warmth. And also blood is flowing into all these capillaries, peripheries. You feel little bit of tingling, itching kind of sensation. And in some people, you see blushing because blood vessels get dilated. Right now, I am going through it. You may not see blushing because I'm not that fair, but I do feel, little bit of itching here. I just took niacin supplement. Once in a while, I take this because niacin come from legumes and lentils at higher concentration. Like if you take, regularly legumes and lentils, it has got niacin. Greens, green leaf vegetable does have, but not as much as legumes and lentils. So as and when, if you get a chance, try to use legumes and lentils because it has minerals as well. All the zinc, magnesium, selenium, manganese, and these things which you need. So it is good idea to use them once in a while so that you get those supplement, nutrients. Anyway, so otherwise just take niacin. It's there in the pharmacy. It's over the counter. Okay. So anyway, that's the niacin, NAD. Remember that. Coming to the very first substrate level phosphorylation in glycolysis. You know what is the meaning of substrate level phosphorylation now? When the substrate is converted to product, there is a generation of ATP. There is a phosphorylation of ADP with PI to make ATP. Right? So your previous product, previous reaction is 1,3-bisphosphoglycerate. It had that high energy phosphate bond, and you're going to remove that now. You just take that high energy phosphate bond from the first carbon and you incorporate that. You're going to take this phosphate from the first carbon. That phosphate is incorporated with ADP, and that's how you generate ATP here. So enzyme has to transfer that. The enzyme has to transfer phosphate from the first carbon to the ADP in the cytoplasm. So any enzyme that transfers phosphate is a kinase. And that's why the enzyme name here is phosphoglycerate kinase. Okay. So why it is written phosphoglycerate? Because it is transferring phosphate from the molecule 1,3-bisphosphoglycerate. That's why they simply written it as phosphoglycerate kinase. So we got ATP in the cytoplasm, not in the electron transport chain. This is in cytoplasm. That's why this is called substrate level phosphorylation, generating a molecule of ATP. And as you all know, from one glucose, you are getting two molecules, such molecules. So that means, I'm sorry, from one glucose, you're going to get two 1,3-bisphosphoglycerate. That means you're going to get two ATPs at this stage. Is it not? Every time we have to say 2 times. Whatever you see here, it is 2 times. Previously, you have seen that. Okay. So coming to what is the mechanism of arsenate that will have an effect on glycolysis? Although briefly I explained you about this in the previous slides. So arsenate competes with inorganic phosphate and hydrolyze glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate without making NADH plus and 1,3-bisphosphoglycerate molecule. What it does is, whenever arsenate is present in the cytoplasm, so that arsenate is going to compete with this inorganic phosphate, because see, arsenate is written here, arsenate, don't worry about that. It's a valency thing. So glyceraldehyde 3-phosphate, as generally, it will be going into 1,3-bisphosphoglycerate, it will make NADH, and later it's going to make ATP, and then you get 3-phosphoglycerate. This is what we have seen, right? So in the presence of arsenate, what happens is, so you don't conduct this reaction, that means you don't make 1,3-bisphosphoglycerate, that means you don't make ATPs. But what happens is, your glyceraldehyde 3-phosphate, it is directly converted into 3-phosphoglycerate. It is simply bypassing a reaction. As you see, it is bypassing one of the reaction down there. It is not allowing 1,3-bisphosphoglycerate to be made. So it simply bring that glyceraldehyde 3-phosphate directly into 3-phosphoglycerate. That means you don't make NADH, you don't make ATP. So if you don't make NADH, that means you're losing 2.5 ATP, right? If you are not making 1,3-bisphosphoglycerate, that means you are losing those ATPs at substrate level phosphorylation. That is I have written here. So it is without making NADH and 1,3-bisphosphoglycerate, while allowing glycolysis to continue, arsenate causes energy deprivation in cells dependent on glycolysis. If the cell is heavily dependent on glycolysis for ATP generation, those cells will be severely affected because you are bypassing the reaction, not allowing energy to be generated. That's how arsenate works on glycolysis. Make sure you know this, how the bypass occurs. So it's not like it's totally inhibiting glycolysis. It is allowing glycolysis to go on, but without making ATPs at this stage. Is it clear? Okay. So that is the point here.